So, today I'm going to discuss the other part of your flower encoding genes for the flower development, how the flower whole or the layers of the flower developed with the help of the floral organ identity genes. Let us see. So, let's get started now. So, if I say about the flower, a complete flower a complete flower has four whorls. Okay, a complete flower has four whorls, or you can say four layers. And what are those four layers? Sepals, petals, stamens, which are the male reproductive part of the flower, and pistil, which are the female reproductive parts. If I see from the base to the top, then this type of arrangement of the flower occurs, right? Now this whole wholes of the flower, which are the sepals, petals, stamens, and pistil, these are promoted by the floral organ identity genes. So, with the help of floral organ identity genes like Apitella 1, Apitella 2, Apitella 3, Pistillata, and Agamus gene. These five floral organ identity genes, they interact with each other and form the layers of the flower. Okay. Okay. Now here I have tried to discuss with you with the help of a diagram, with the help of a concentric diagram, and this diagram actually represents the four whorls of the flower. Okay, with the help of the this diagram, this is called a concentric diagram. With the help of this diagram, I have discussed about the four whorls of the flower. Now if I see from the base to the top of the flower, then the first organ that we see is the S. Well, S is the sepals. Okay, sepal is also known as the calyx, Calyx, and the general function of the sepals is to protect the developing flower. So this is the first whorl. That's why I have written S1. Sepal is the first whorl or the first layer of the flower. If we look from the base, what comes after the sepal? After the sepal, we find beautiful colored patterns of the flower that we call them as petals. Well, here we have represented petals as P, capital P. So petals are the second whorl or the second layer of the flower. Then comes the main part. Well, sepals and petals, if I see, these sepals and petals are called as accessory floral whorls because they normally are the helping parts of the flower for conducting pollination and to protect the flower only. Whereas the rest of the two parts which are found on the top, the stamens and the carpel, these are the main parts of the flower, the main reproductive parts of the flower. Well, the stamens, if I give a different color, as written by ST here, stamen is the male reproductive part organ of the flower, and it is also known as androecium, Androecium. It is also stamen is also known as the androecium, and stamen consists of anther and filament. If you look at the notes, you will see the most top part of the flower is the uh, carpel, okay, denoted by C. carpel is the fourth number old, the fourth layer of the flower, which is found at the top. Okay, carpel is the female reproductive part of the flower, and it is also called as gynoecium. Carpel is also known as gynoecium, right? And carpel consists of stigma, stele, and ovary. Okay, stamen and carpel both are essential floral whorls, whereas sepal and petal are the accessory floral whorls. Okay, now we have studied of these floral diagrams, the genes responsible for the floral formation, the flower formation, which are called as floral identity genes. Floral organ identity genes are actually studied in this organism, Arabidopsis thaliana, which is a long day plant, and it is a member of a family called Brassiaceae, which is of mustard family and is widely used as a genetic model in plants. Okay, so this all concept of identifying the floral organ identity genes, the epitela 1, epitela 2, epitela 3, pistillata, and agamus, these are all identified in Arabidopsis thaliana plant, and based on which we have scientists have revealed the genes and how the arrangement of flower occurs. They have studied. Now there are 5 floral organ identity genes, as I have mentioned with you, right? As I have mentioned with you, and those genes are epitela 1, epitela 2, epitela 3, pistillata, and agamus. Floral organ identity genes also have another name known as floral homeotic genes. So the other name of floral organ identity genes is also called as floral homeotic genes. all of them all of them except your epitela 2 contains the same DNA binding domain, madbox. So the floral organ identity genes are also called as floral homeotic genes. That's good. And all of these floral organ identity genes have, except AP2, the other 4 floral organ identity genes have a DNA binding domain. Domain is the essential region, and the name of that domain is called MAD box. MADS box, MADS. So these 4 genes, which are epitela 1, epitela 3, pistillata, and agamus, these 4 genes have MAD box sequence that encode protein that contains the MADS box binding domain, DNA binding domain. Now what is MADS box? What is MADS box? Some type of conserved sequence found within these floral organ identity genes. Let us see, let us study, because it has been described in your syllabus that to study about the MADS box. So the floral organ identity genes contain this MADS box DNA binding motif. Okay, what is MADS box? Let us see. So I have said repeatedly, he has been written here, except AP2, except epitela 2, the 4 floral organ identity genes have MAD box sequence. When these genes encode proteins, they will have the same DNA binding domain called as MADS box DNA binding domain. What is MADS box? Let us see. MAD box is a conserved sequence motif What do you mean conserved? That means it is found. It is a unique sequence which is found among the species. So Matchbox is a conserved sequence motif. What is motif? Motif is a nucleotide or amino acid sequence pattern which is widespread and usually assumed to be related to the biological function of the macromolecule. So Matchbox, it is a conserved sequence motif, means it consists of a sequence which is assumed to be related to the biological function of that particular protein. Here the protein are the all the floral organ identity gene proteins. Okay, now those genes which contains this motif are called Matchbox gene family. So those genes who have this type of Matchbox DNA sequence, they are called as Matchbox gene family. And the Matchbox encodes DNA binding MADS domain. Okay. And what is the special feature of this Matchbox sequence is that if you see the sequence of the Matchbox, which is found within the all the floral organ identity genes, these Matchbox domain bind to the DNA sequence of high similarity to the motif CC either A or T 6 times GG is termed as CARG box. Now normally MADS domain, that means, again I repeat slowly. So it means to say that these floral organ identity genes have a Matchbox sequence, right? When these floral organ identity genes will encode protein, will encode enzyme, they will have this Matchbox motif, Matchbox domain, and they will bind to the specific DNA sequence. Now where it will bind? That sequence has been mentioned here. CC, A or T 6 times, then GG, right? This sequence is also called as CARG box. That means within this, if a DNA contains this sequence, then the MADS box binding domain will bind. Whoever it is, it will bind at that site related with this sequence. Okay, related with this sequence. Okay, so what's the main purpose of this Matchbox is that Matchbox domain proteins are generally your transcription factors. So the Matchbox domain proteins are your transcription factor. So those proteins that contain Matchbox sequence, they are actually transcription factors. So here the floral organ identity genes are actually a type of transcription factor which triggers the transcription of the genes responsible for the flower development, responsible for the layers of the flower, and they will bind to this type of certain sequence with the help of MADS domains within them, right? Now Matchbox has a variety of function. So I think you have understood this part. I will try to recapitulate. I will try to revise this part again. So I have said about Matchbox that all the floral organ identity genes except your AP2, except epiteller 2, the other floral identity genes have this Matchbox. Well, Matchbox is a DNA binding motif and it binds to this type of sequence called as CARG box. Generally, Matchbox encode the proteins, those who have that Matchbox motif, the proteins that those proteins that have this Matchbox domain, they encode transcription factor, right? And Matchbox gene have a variety of function, like in animals, as you can see here, Matchbox gene are involved in muscle development and cell proliferation and differentiation. So the first function that we found in animals, for example, here, well, we should not describe here about animals, but at least I try to describe, because otherwise you will not able to understand that it has vast functions. Matchbox have vast functions. Like here, matchbox gene, that means the genes, those genes that have this matchbox motif. In case of animals, matchbox genes are involved in muscle development. That means in animal cells, those genes that have matchbox sequence, they help in muscle development. They help in cell proliferation and differentiation. Okay, I think you have understood. Then in fungi also, this type of matchbox genes can also be observed, right? Like in fungi, it ranges from pheromone response to arginine metabolism. So in fungi, it ranges from means it has many functions, like those gene that have matchbox sequence, they can act as pheromone. Now what is pheromone? What is pheromone? Pheromone response to arginine metabolism. Okay, that means what is pheromone? Pheromone is actually a chemical that an organism produce normally, which changes the behavior of another organism of the same species. If I say about pheromone, pheromone is a chemical that an organism produce, which changes the behavior of the another organism of the same species. Pheromones are similar to hormones, but they work outside the body instead of inside. So they here, in case of fungi, the genes, those who have this matchbox sequence, they can act as pheromone. They can act as pheromone response to arginine metabolism. Okay, okay. Like for example, I have given more examples of pheromones here just for knowledge. Like androsterol, androstenol. Sorry, androstenol. It is a common pheromone which is produced by the female mammals. Okay, which is like a musk-like odor, which is found in the sweat, in the urine, in the plasma, in the saliva, which triggers the feeling of attraction and security in women. Okay, so it's work outside actually, like a hormone, like a hormone, but it works outside the body. So here, what I am trying to say about Matchbox, that in it has variety of functions. Like in flower development, Matchbox plays an important role. It acts as a transcription factor. Okay, but that means those genes that have Matchbox sequence will encode protein, and thus they will have that domain, matchbox binding domain, which will bind to the certain DNA region called as CARG box, which I have said just now little bit ago. Now depending on that, they will trigger certain genes to transcribe for the development of flowers. In case of animals, like here I have said that those genes that have matchbox sequence, they help in muscle development. In case of fungi, it function as pheromone pheromone response to arginine metabolism. Okay, okay. So these pheromones plays an important role in humans, in other animals, even in fungal species also. It plays a critical role in choreographing interactions, which are the sequence of steps between the mating partners during the program of sexual reproduction. Okay. Now come to the plants. In plants, these matchbox genes are involved in controlling all the major aspects of development, including the male and female gametes, embryo and seed development, as well as root, flower, and fruit development. That's why matchbox genes are found everywhere, also in plants. Okay, so this matchbox gene, the genes which encode the genes which have this matchbox sequence, they are involved in many aspects of development in flowers, in plants, like male and female gametophyte development, embryo and seed development, as well as root, flower, and fruit development. Okay, gametophyte are the stages which produce the sex cells of the plants, and algae that undergoes alternation of generations. Now, so various function are there of those genes that have this matchbox sequence. Now, some matchbox genes of flowering plant have homeotic function. Homeotic function means those genes that control the pattern of body formation, okay, during early embryonic development of organisms. And this homeotic function means these genes, the matchbox genes here, will encode proteins called transcription factor, which will direct the cells to form various parts, organs of the body. So, matchbox gene of flowering plant have homeotic function, which will control all the development of the plant parts, like the Hox gene of animals. Just like the Hox gene of animals, they determine the they determine the how development of the body parts will occur. Similarly, Matchbox gene. So, as I have said here, that most of the Matchbox genes of flowering plants have homeotic function. It means that these genes actually control the pattern of body formation during your early embryonic development of organisms. And actually, they encode proteins which are transcription factors, which directs the cell to form various parts of the body. So here, just like the Hox genes in animals, the matchbox gene of flowering plants have homeotic function. The floral homeotic matchbox genes like agamas and deficience participate in determination of floral organ identity according to the ABC model of flower development. So here it has been said that these two genes, deficience, agamas and deficience, they participate in determining the floral organ identity according to the ABC model of flower development. Now, we will discuss about the ABC model of flower development just in a moment, but we have to see other matchbox genes which participate. So, all the floral organ identity genes have the matchbox sequence, which will ultimately, when they will form protein, they will have that matchbox but DNA binding domain, and all of them encode transcription factors which will direct the cells to form various floral parts of the plant. Another function of matchbox gene is flowering time determination. Now, within Arabidopsis thaliana, there are some genes that have matchbox sequence like SOC1. We have seen SOC1 when we are talking about the different ways of flower development, and flowering locus C, also called as FLC, have been shown to have an important role in the integration of molecular flowering time pathways. So there are also some other genes other than your floral organ identity genes that have the matchbox sequence like your SOC1 and flowering locus C genes, which have been shown to have an important role in integration of molecular flowering time pathways. Well, I don't think that SOC1 full form has been mentioned somewhere. Okay, I have to see if I get that name. Okay, I have get that name. SOC1 full form is suppressor of overexpression of CO1. Suppressor of overexpression of CO1. CO1 is actually contents which actually involve during photoperiod. So this SOC1 and the flowering locus C, this gene contain the matchbox sequence, and these genes are essential for correct timing of flowering and helps to ensure that fertilization occurs at the time of maximal reproductive potential. Okay, so matchbox have different functions. They help in flower development, and most of the matchbox genes have homeotic function like agamas, like deficiency. Then it also determine the correct timing of the flower development like the SOC1 and flowering locus C, which ensures that fertilization occurs at maximal reproductive potential of the plant. Now if I see the structure of matchbox, I have not got a diagram of matchbox sequence, and that's not required now for your undergraduate portion. Matchbox protein structure is characterized by four domains. So there are four essential regions. N-terminal end, which is the amino terminal end, which has the highly conserved MADS DNA binding domain. There is some intervening and keratin-like domains which are involved in specific protein-protein interactions. And the third domain is the carboxy terminal domain, which is highly variable and involved in transcriptional activation and assemblage of heterodimers and multimeric protein complex. So, Madbox protein structure, if I see those protein who have this Madbox binding motif, they have four domains. First, they have amino terminal end, and after that, they have a highly conserved Madbox DNA binding domain, then an intervening sequence and a keratin-like domains which are involved in specific protein-protein interaction, then carboxy terminal domain which is highly variable and is involved in transcriptional activation and assemblage of heterodimers and multimeric protein complexes. So I have discussed about the matchbox. Now let us discuss about the floral homeotic genes or the floral organ identity genes. Well, it has been mentioned here that floral homeotic genes are also called as matchbox genes because they have matchbox sequence. Floral organ identity genes are classified into three classes as I have discussed in previous lecture: class A genes, class B genes, and class C genes. Apitella 1 and Apitella 2 fall under the category of class A gene. Apitella 3 and Pistillata fall under the category of class B gene, and Agamas fall under class C gene. Well, class A gene, these genes promote the development of sepal and petal. So, Apitella 1 and Apitella 2, they promote the development of sepals and petals. Apitella 3 and Pistillata, these genes promote the development of petal and stamens. Okay, so these two genes fall under the category of class B genes. They promote the development of petal and stamen, and class C gene promotes the development of carpel and stamen. So as I have said that class A genes promote the development of sepal and petal, which are the accessory parts of the flower. Class B gene helps in the production or promote the production of petals and stamen. Class C gene promotes the development of carpel and stamen. So these all genes directly control the floral identity and helps in making the flower or floral part of this. Now let us see with the help of this concentric floral diagram to understand about this expression of floral organ identity genes. Okay, so with the help of these floral organ identity genes, let us see what we can conclude from this diagram. Well, I have said the first ole is your sepal, second ole is your petal, third ole is your stamen, and fourth ole is your carpel. Sepal is promoted by the class A gene. And what are the class A gene? Would you guess what are the class A gene? I have already discussed with you. Class A genes are the Apitella 1 and Apitella 2. These two genes are class A gene. They promote the development of sepal as well as petal. For the development of petal, it is promoted by class A gene as well as class B gene. So both class A gene and B gene are required. Well, we know the class A gene, Apitella 1 and 2. What are the examples of class B gene? Apitella 3 and Pistillata. These are the class B genes. So class B gene interact with class A gene to form the petals. Okay, the second ole. The third ole, which is the stamen, the main male reproductive part of the flower, it is promoted by class B gene and class C gene. So interaction between class B and class C gene promote the development of stamen, right? Class B gene I have already discussed, Apitella 3 and Pistillata. Class C gene are agamas, which is AG. And for the fourth ole, only the carpel, for only carpel development we require agamas gene, which is class C gene. This is the generalized gene and gene interaction which I have discussed with you. Now, from this diagram, if I can see that either A and B are interacting with one another or B and C. A and C are not interacting with one another, so they are antagonistic with each other. It simply means that if A gene is mutated, then C gene is activated or expressed. If C gene is mutated, A gene will be activated. Both of them will not together express. Okay, I think I have mentioned to you in the right way. So here we can see only A gene is promoting the development, only the A class gene is promoting the development of sepal. A gene and B gene are interacting with one another to form the petal formation. That's why I have written in this way. B gene and C gene, better to say class B gene and class C gene, they are interacting with one another to form the stamen, and only the C gene, class C gene, are forming the development of carpel. But A and C are antagonistic with each other. Okay. Now, if mutation occurs in one of these classes of genes, then what will happen? Let us see one by one. So I have drawn a chart here, but I will not discuss here. Better to discuss from this side. Okay. So this is the wild type or normal. For the development of sepal, class A gene is required. for the development of petal, both A and B gene is required. Class A and class B gene is required. For the development of stamen, class B gene and class C gene is required, and for the development of carpel, only class C gene is required. I think you have understood. Now, the first case, if class A gene is mutated, then what will happen? Well, I have said that class A gene and class C gene are antagonistic with each other. They are mutually antagonistic. So, in that place, if A gene is mutated, the class C gene will take place in the place of class A gene. So, in the place of class A gene, as class A gene is mutated, class C gene will take place in that site. Thus, your first OL will be carpel, because class C gene promotes the development of what? Carpel and stamen. For development of stamen, B plus C. So the second OL will be stamen. Third OL will be also stamen, and the fourth OL will be carpel. So there will be no formation of sepals and petals if your class A gene is mutated, because in that place class C gene comes, and the first OL, instead of having sepals, what happened? It formed carpel, because A gene is mutated. So C gene takes place in that site. Second OL, or the second layer, will be your stamen. Instead of petal, stamen occurs, because class B gene interacts with class A to form your petal. But here A gene is mutated, so in that place C gene comes, B and C interact with one another to form stamen. Then again stamen occurs in the third OL, and in the fourth OL carpel occurs. So no formation of petals or no formation of petals or sepals occur if class A gene is mutated. Let us see the second case. The second case, suppose class B gene is mutated, then what will happen? If class B gene is mutated, then what will happen? Well, B interacts with A to form the petal, and B interacts with C to form the stamen. So there will be no petal or stamen formation. So the first OL of the flower will be sepal, as A gene is normal. The second OL will be also sepal, because no B gene is there. Class B gene is mutated, so only A gene is expressing to form sepal. The third OL will be carpel, because as no B gene is there, it is mutated, so it will not able to interact with C to form stamen. And the fourth OL will be carpel again. So if class B gene is mutated, then there will be no petal or and stamen formation. Okay, and stamen formation. I think you have understood what I have said. Third case, if class C gene is mutated, then what will happen? Well, in that place class A gene will take place. So one thing clear, carpel will not develop, male flower will occur. Not only the carpel, but also the stamen will also not develop, because class B gene interact with class C to form the stamen. So stamen as well as your carpel will not develop. Permanently sterilized flower will occur. Only for show the flower forms here. First OL is sepal, because A is normal. Second OL petal, okay, A interact with B. The third OL, where B used to be interact with C, but C is mutated, so in that place A class A gene comes, again petal formation occur, and the fourth ole in place of C, as C is mutated, A gene is activated now. It took the place of class C gene and forms sepal again. So there will be no formation of stamen or carpel. So entirely in this case, only the accessory part of the flower develops. The main part of the flower, which is needed for the carpels and stamens, are not formed. Okay. Now let us see the fourth case. So total there are 6 cases. If both class A and class B gene is mutated, then what will happen? Generally, if class A gene and class B gene is mutated, then only the C gene is there, na? So in the all the four layers of the flower, carpel will develop only. It will be not attractive. All the all the four floral whorls will develop carpels if the A and B gene gets mutated. Now, if B and C genes are mutated, then all the four whorls will have sepals in them, completely sterilized. In the above case also, it was sterilized flower because no reproductive organ has been formed in the flower. And in the last case, if all the three genes are mutated, then there will be no sepal, no petal, no stamen, carpel formation will occur. And that type of flower is called pseudo flower, which have a green-like appearance, leaf-like appearance, but looks like a flower, but it's called a pseudo flower. Pseudo means false. Okay. So this was all about the today's lecture. I hope you have understood what I have tried to mean. Okay, just go through these diagrams. I hope you will able to understand after going through the diagrams and the video link that I have shared with you previously. Just go through it. Okay.